We are going to continue talking about analyzing DNA and genetics by discussing DNA cloning and characterization. So what is DNA cloning? It's being able to make multiple copies of a certain DNA sequence. There's a large quantity of individual genes in pure form. These are required to study the gene structure and function. So if you wanted to study a gene, we cannot only use a single copy of that gene simply because we won't be able to manipulate it or detect it or even see it. So DNA cloning permits researchers to prepare large numbers of identical DNA molecules, so multiple copies of a certain molecule. These molecules usually code for a single gene. The key to cloning a DNA fragment is to link it to a vector DNA. This vector DNA molecule can replicate within a host cell. So this DNA segment is inserted into a vector. That vector can make multiple copies of itself in a certain host cell. This could allow us to generate large numbers of DNA fragments by replicating the cell itself as well. So not only does the DNA segment in the vector replicate itself when the vector is replicated, but the vector is more replicated when the cells themselves divide into more than a single cell. So the process for DNA cloning starts with the target vector. So this is a gene sequence that we are interested in. We insert it into a DNA fragment. This gives us what we call a recombinant DNA. Then this recombinant DNA is going to replicate itself within the host cell. So it's going to make multiple copies of itself within the cell. Also, the cell itself is going to make multiple copies of itself by dividing through mitosis. After multiple cells are present with multiple copies of the recombinant DNA, the resulting DNA or the target vector DNA here is going to be isolated, sequenced, and manipulated to whatever target we want to use it for. So in order to cut out and insert and stick the DNA sequence that we want to the vector that we discussed, we need specific enzymes. The enzymes that cut the DNA are called restriction enzymes. These restriction enzymes are endonucleases. They are usually produced by bacteria. This bacteria has different endonucleases, each of which recognizes a sequence of 4 to 8 base pairs. These sequences are called restriction sites, and this restriction enzyme, once it recognizes the restriction site, it cleaves both DNA strands at these sites. So we cut out the sequence that we are interested in. If we know the sequence, we know where the restrictions, what available restriction sites we have. So we look for a corresponding restriction enzyme. We add it to the DNA, and the restriction enzyme cuts out the sequence that we want. So if we want to cut out that sequence, the restriction enzyme has to cut both ends of the gene that we are interested in, or we're going to have to add more than one restriction enzyme, each for each site we want to cut. The enzymes that stick the DNA together, we have mentioned them before, are called ligases, and there are ligases produced by bacteria. These ligases connect DNA sequences one to the other. As we just mentioned, restriction enzymes cut double-stranded DNA at a certain location called a restriction site. This is an example where the restriction enzyme called E-core-I cuts double-stranded DNA at a sequence similar to this one. And if you notice, it cuts it each strand at a different location. If you notice, it's a mirror image location. However, what's interesting is that these single-stranded DNA ends stay outside and allow a complementary sequence that the restriction enzyme cut at a different location to complement it and to stick to it. These single-stranded DNA ends are called sticky ends. If in the case there's what we call a blunt end, where a restriction enzyme cuts the double-stranded DNA, both strands at the same location. So if it cuts it in a way that is similar to this, a straight line, this is called a blunt end, not a sticky end, and that would make it difficult for DNA to reassemble itself with a complementary sequence similar to a sticky end. However, both types of restriction enzymes are present. So for each restriction enzyme bacteria, there's a modification enzyme which protects a bacteria's own DNA from cleavage by modifying at or near the cleavage site. So the purpose of restriction enzymes in nature is not to use them to replicate DNA, but for bacteria to protect itself from foreign DNA, so for example from viruses, and it protects itself by these restriction enzymes cutting up any foreign DNA that inserts the bacterium. So for the bacteria to protect itself from the restriction enzyme that it produces, so that the restriction enzyme would only cut up foreign DNA, what it does that the bacteria modifies its own DNA to prevent it from being cut by its own restriction enzymes. That modification usually involves adding a methyl group at the restriction site. The presence of this methyl group would prevent the restriction enzyme from cutting the DNA. So the methylation of the restriction site in a bacteria is a defensive mechanism against foreign species and their own DNA. Single-stranded regions, sticky ends, can base pair with others cut by the same restriction enzyme, because if you remember I showed you it's a mirror image. So if we cut a sticky end, the complementary sequence can be found by any other sticky end cut by the same restriction enzyme. The frequency with which a restriction enzyme cuts DNA and the average size of the resulting fragment is largely dependent on the site of the recognition site. So if the recognition site is larger, the restriction enzyme takes longer to cut it, and the fragment, of course, that results from it is larger. Usually, the length of the restriction enzyme is about 4 base pairs of that of the restriction site. So these are examples of different restriction enzymes. This is just to show you how different enzymes are going to cut at different restriction sites. So this one results with a sticky end. This one also cuts before the G and after D, also results in a sticky end, as well as this one and this one. This one results with a blunt end. It cuts it right straight ahead away, and as we just mentioned, this one has the largest restriction site. So as we just mentioned, because it has the larger restriction site, its cutting of the DNA is going to be less frequent and less aggressive. The vectors that we mentioned at the beginning of the slide sets are DNA sequences in which we insert DNA fragments. Here we use restriction enzymes to cut both the vector and the DNA fragment of interest. These restriction enzymes are going to result with either a blunt or a sticky end, depending on what the restriction enzymes and DNA ligases are available. As I mentioned earlier, ligases are usually used to covalently bind the ends of the restriction fragments and the vectors. If you remember when we talked before about ligases, we mentioned how they are present in the process of DNA replication and in DNA repair. So they are there for these purposes. However, we use them in biotechnology in order to covalently bind the ends of different DNA fragments and vectors. So if we have a vector DNA sequence that we can use, we have a genomic DNA fragment of interest. So what we want to do is we need to attach this fragment to the vector DNA. So what we do is we cut both the DNA fragment and the vector DNA with the same restriction enzyme. This would result us with sticky ends that complement one another. So once these two are mixed together in the same vials, they are going to naturally base pair. And once they base pair, these are, as you know, base pairing is the secondary bond. It's not a permanent bond. What happens is that we add T4 DNA ligase, which is a type of purified ligase, and the ligase covalently attaches the loose ends of the complementary sequence with between the fragment and the vector DNA, and then we're going to have a whole sequence. This is the sequence we are interested in, and this is the vector DNA that we are going to use for replication, so we can make multiple copies of the genomic DNA fragment of interest. One type of vector DNA are plasmid vectors. These plasmid vectors are circular. They are double-stranded DNA molecules. They are separate from a cell's chromosomal DNA, and they are usually present in bacteria. You cannot find plasmid vectors in eukaryotes. A very common plasmid vector is that of the bacteria E. coli. A lot of these vectors have been engineered to remove unneeded regions and making plasmid vectors that would help replicate DNA fragments of interest. So for a plasmid vector to be useful, it has to have the following components. These, as I mentioned, can be engineered by genetic engineers. The vector has to have what we call a replication origin. If you remember when we talked about DNA replication, we mentioned where the point where DNA starts to make a bubble so it can start replicating itself and make two replication forks at that bubble. That starts at the replication origin, and it's usually 50 to 100 base pairs. In addition to a replication origin for the plasmid vector to replicate itself, we need a marker that permits selection, and I'll describe that in a bit. Afterwards, we need a region where we can insert the exogenous DNA fragment that we are interested in replicating. So this is an example of a plasmid vector. This plasmid vector has three components. The first one, as we mentioned, is the origin of replication. The second part is the region where exogenous DNA can be inserted. So this region is called the polylinker, and it's called that because it has multiple locations for different restriction enzymes to be cut. So these all are names of restriction enzymes, HIN3, SPH1, BAMH1. These all are different restriction enzymes. So depending on the sequence that we want to insert, we use the corresponding restriction enzyme and cut both the plasmid vector and the fragment that we want to insert in that vector with the same restriction enzyme, preferably with a restriction enzyme that results with a sticky end. The final component of the plasmid vector is a selection mechanism, as we just mentioned. And the reason for this is that we need to know if a bacteria has taken up this vector or not. So once we have inserted the sequence of interest, we are going to make E. coli bacteria take up this vector so that this vector can replicate itself inside the bacteria, and while the bacteria also divides into multiple copies of itself. So we add this vector to the E. coli. However, we need to select E. coli cells that have the vector versus E. coli cells that did not take up the vector. The way the bacteria takes up the vector is through a mechanism we call heat shock. So we add these vectors to a vial that has the E. coli bacteria present in it, and we raise the temperature. This provides the bacteria with what we call a heat shock and cause the bacteria to take up these plasmid vectors. However, we cannot guarantee that all the bacteria present in the vial has taken up the plasmid vector. In order to do so, we need the presence of this gene. This example here, the gene has ampicillin resistance. So it provides the bacteria that takes up this vector with resistance to the antibiotic ampicillin. So if after the heat shock we plate the bacteria on a dish that has ampicillin on it, only the bacteria that has ampicillin resistance will survive. In other words, only the bacteria that has taken up the plasmid vector would survive, making it a selection mechanism for the bacteria that has taken up the vector. So this is the full process of replicating a DNA fragment of interest in an E. coli cell. So as we mentioned, we have a DNA fragment to be cloned, and we have the plasmid vector with its polylinker and its resistance mechanism. So as I just mentioned, we cut both the DNA fragment and the plasmid vector using the same restriction enzyme. Then we insert the DNA fragment in the plasmid vector, and we add the enzyme ligase, so it would result with a recombinant plasmid that has the DNA fragment to be cloned as part of it. So afterwards we mix the plasmid with the bacteria E. coli in the presence of calcium chloride and a heat pulse, as I mentioned, that to provide the bacteria with a heat shock. This causes the bacteria to take up the recombinant plasmid. Then the bacteria is cultured on agar plates containing ampicillin. Cells that don't have the plasmid die because they were subjected to ampicillin, and they do not make proteins that are resistant to ampicillin because they don't have the genetic code for it. However, cells that are transformed, the meaning of a transformed cell is that one that has taken up foreign DNA. The transformed cell is going to survive the ampicillin plated dish because it is going the plasmid in it is going to provide the cell with resistance to ampicillin. Afterward, the plasmid is going to replicate itself inside the cell, making multiple copies of the DNA fragment of interest, and afterwards the cell itself is going to multiply using mitosis, and all these cells are going to have multiple copies of the recombinant plasmid here. So we're going to have multiple copies of the DNA fragment that we want to clone. This brings us to the topic of cDNA libraries. cDNA stands for the word complementary DNA. These DNAs are synthesized using an enzyme called reverse transcriptase. And reverse transcriptase literally reverse transcribes mRNA. If you remember, transcription involves converting a DNA sequence into an RNA sequence. So reverse transcription is the exact opposite of that, where RNA is converted into a DNA sequence. The reason why we want to use RNA in order to get a DNA fragment and not to extract a DNA fragment directly from the nucleus is, as you remember, DNA in the nucleus has untranslated regions that are not of interest if we want to synthesize a certain protein or if we want to know what the cell is synthesizing. So we start from the mRNA, use reverse transcriptase, and that reverse transcriptase results in multiple DNA copies. These would cause the production of a genomic library that has all the DNA sequences in the genome for a particular species. So we can collect all the mRNA that's present in the body, reverse transcribe it, and that would give us the genomic library, because if you remember, the genome has all the genes, and each gene has the code for a certain protein that is synthesized by the body. And the protein gene does not include untranslated regions. So in order to prepare the cDNA, as I mentioned, total mRNA is isolated from a certain cell type or tissue of interest. If you remember, there's one common sequence in all mRNA, and that's the poly A tail at the end of the mRNA. This will help us to extract mRNA from a cell lysate. A cell lysate is a mixture of cells whose membranes has been torn apart. So we can extract the mRNA by letting the mRNA anneal to chromatography beads that have poly T tail, polythymine tails that are attached to them, because a poly A tail is going to attach to a poly T tail. So if we add the cell lysate to a chromatography column that has a poly T tail bound to its bead, the mRNA is going to bind to these poly T tails, and then we can elute it and extract the mRNA. After we obtain the mRNA, we use the enzyme reverse transcriptase to synthesize the strand of DNA that is complementary to the mRNA, starting from the primer, which is the T-tail primer. So the resulting cDNA mRNA hybrid molecules are then converted to double-stranded DNA. To prepare this double-stranded DNA for cloning short double-stranded DNA molecules containing the recognition site for the restriction enzyme that we know specifically are ligated to both ends of a cDNA using the T4DNA ligase. Ligase, as we mentioned, can join both blunt ends and sticky ends that are treated with different restriction enzymes. So this is the process of cDNA preparation. We first extract the mRNA present by allowing mRNA with its poly A tail to hybridize to the oligo DT primer. Once it's extracted, we want to make copies to convert the mRNA into a double-stranded DNA. So we obtain the oligo DT primer. This DT primer is going to adhere to the poly A tail through simple base pairing. And then here we add the enzyme reverse transcriptase that transcribes the RNA into a cDNA or a complementary DNA sequence. So we have now a hybrid RNA-DNA double strand. We only need the DNA strand after this step, so we remove the RNA by adding a certain chemical, and this would cause and add a poly G tail to the other end of the cDNA. The poly G tail is going to be hybridized to a poly C tail, and then we're going to get to synthesize the complementary strand DNA using a DNA polymerase and not reverse transcriptase as we did in the first step. So we synthesize the complementary strand, and then in order to prevent restriction enzymes from cutting certain sequences within the mRNA sequence, we methylate the site where if a restriction enzyme can cut in the middle of the sequence, and then we ligate the ends of this DNA double strand the DNA to a two equal linkers. Afterwards, we add a restriction enzyme that would cut these linkers and cut a plasmid vector with the same restriction enzyme, creating complementary sticky ends, which would allow the formation of a recombinant plasmid that contains the original sequence that we obtained from the mRNA. And then we transform E. coli with this recombinant plasmid vector and go through the same process that we discussed a couple of slides ago. One reason we want multiple copies of a certain DNA strand is to be able to hybridize it to fabricate oligonucleotide probes. So this is a method to identify a DNA sequence, and this takes place through the ability of complementary single-stranded DNA or RNA molecules to associate with each other via base pairing. So if I have a sequence that I know, if I have a certain sequence that I know, this sequence would only bind to its complementary sequence. If we get a different sequence, it's not going to bind to it. It's not going to base pair to it. So it only binds to a complementary sequence. So if we have a bunch of DNA whose sequences we don't know, and we have a plate, and that plate has different strands whose sequence we know, we can check where the unknown DNA binds, and we know that it would only bind to its complementary sequence. So that way we can identify the DNA sequences or the RNA sequences that we are not sure of their sequence. So this is the process of the hybridization of oligonucleotides. So we have single stranded DNA. We use a filter, and then we incubate this filter with we incubate the DNA that we don't know with complementary DNA. The labeled DNA is washed away, so only the DNA that is bound to its complementary sequence would remain. This is the purpose of the washing step. So since we know the sequences that we added, we know that the complementary sequence are the ones that are going to bind to it, and we know it's a complementary sequence, so we can guess what the sequence is. This brings us to the discussion of shuttle vectors. Shuttle vector is similar to a regular vector, however the main difference is its capability of propagating or making copies of itself in two different types of cells, in two different hosts. It has the element components that we have seen in the E. coli vector. However, it has other components that would allow it to replicate in a eukaryotic cell. These include the presence of an autonomously replicating sequence, an ARS. This functions as the origin of replication of DNA, however not in E. coli but in yeast. There's also a component that's called a yeast centromere. This component allows faithful segregation of the plasmid once a yeast cell division takes place. And the last gene that is present on a shuttle vector is the yeast gene encoding an enzyme for uracil synthesis. This is also as a selection marker similar to the ampicillin resistance marker that we have previously described. So if you see, a shuttle vector has 6 components. unlike a plasmid vector, so it has double the components of a plasmid vector. We have the origin of replication that we had in a plasmid vector, the ampicillin resistance gene as we did in a plasmid vector, and the polylinker where multiple restriction sites are present. However, we also have the centromere gene, we have the autonomously replicating center, and we have the uracil selection gene. So the shuttle vector is also cut with a specific restriction enzyme, which we also use with the DNA fragment of interest. Then we insert these different vectors that we are interested in within the shuttle vector, and we ligate them to make a recombinant shuttle vector. Then we transform these vectors into an E. coli cell and screen for ampicillin resistance. This would show which cells have taken up the vector and which didn't. Then we allow the vector to make copies of itself and for the E. coli cells to replicate. Afterwards, we isolate the recombinant plasmids from the E. coli colony. Then we use functional complementation, which is something we discussed in the previous slide set, to select for certain genes. If you remember, in the complementation test, we had a temperature sensitive mutant. If you remember, we had the miscible and the immiscible temperature. So we transformed the yeast cells by treating them with LiOAC, PEG, and a heat shock, so not with calcium chloride as we do with the E. coli. Then we plate and incubate the yeast cells at the permissive temperature on a medium that does not have uracil. So cells that have the uracil gene are going to survive, and cells that do not have the uracil genes are going to die, because uracil is an important component of their nutrition. If they are not able to synthesize it using the gene, that would cause the cells to die. So in this uracil gene is the selection method in the yeast cells, where the ampicillin resistance is the selection gene for the E. coli cells. Then we replicate the plate and incubate it at the non-permissive temperature. And as we discussed earlier, only the cells that have a wild-type gene are going to be able to grow, and those that have the mutation won't be able to grow at the non-permissive temperature. So a technique to differentiate between digested DNA strands in order to separate them based on their molecular weight is a familiar technique, gel electrophoresis. We have already discussed gel electrophoresis in detail and in a more complex manner when we discussed gel electrophoresis in proteins. So the same principles that apply to proteins are going to apply to DNA. So usually long DNA strands are cut down into smaller pieces using restriction enzymes. Afterwards, no more processing is needed for DNA, because all DNA has the same structure, unlike proteins, and all DNA has a negative charge, again, unlike proteins. So the DNA is placed in wells in an agarose or a polyacrylamide gel, also similarly to proteins. Once the DNA is placed into the well, at the beginning of the gel, the porous gel, an electric field is applied, and DNA is going to move from the negative side of the gel to the positive side of the gel. Of course, it's going to be easier for smaller particles of DNA to move further along the gel. So they are going to move in the gel at a rate that is inversely proportional to their chain length. So the shorter DNA strands are going to be at the bottom of the gel, while the larger ones are going to travel a shorter distance and be closer to where the DNA has started. After the gel is run for about an hour or so, in order to be able to visualize DNA strands inside the gel, the DNA inside the gel is subjected to auto radiography, so in a kind of an X-ray chamber sort of thing, or the gel can be incubated with a fluorescent dye that is going to bind to the DNA strands and show you the distribution of DNA based on its molecular weight. Again, gel electrophoresis for DNA is simpler because near neutral pH, DNA carries a negative charge, so we don't need to modify its charge. The gel matrix, similarly to proteins, restricts random diffusion, so we need the gel matrix in order to eliminate any other forces on the DNA strands. Smaller molecules usually use polyacrylamide gels because they have smaller pore size, where larger molecules usually use agarose gels that have a larger pore size. DNA a lot of time is bound to the substance ethidium bromide. Ethidium bromide increases its DNA's intrinsic fluorescence and enables us the viewing of DNA inside the gel, whether it's a polyacrylamide or an agarose shell, once we subject it to UV light. A very important DNA amplification process, and something that has become very common in popular culture with the pandemic, is PCR, or polymerase chain reaction. It's called polymerase chain reaction because it involves the enzyme polymerase, and it's called a chain reaction because it's a reaction that takes place in a cycling process, making a chain reaction. So what happens in PCR, which is, as I mentioned, a DNA amplification technique in order to get enough DNA to detect, is we start out by double-stranded DNA. Whether we recognize this DNA or not is not a problem, as long as we have proper primers. At the beginning, we denature the double-stranded DNA by subjecting it to high temperatures. Once it's subjected to high temperatures, present primers in the reaction anneal to their complementary sequences in each strand. Afterwards, the enzyme polymerase is going to extend these two primers as it does in the replication process, because if you remember, in DNA replication we mentioned that DNA polymerase only works when it binds to a short DNA or RNA strands, hence the necessity of the presence of a primer. Afterwards, we started with one strand, then we ended up with two strands. That is the end of the first cycle. Then we denature both strands that are present by heating the mixture again. This is going to cause all four strands to be separate from one another. Then we lower the temperature a little bit in order for the primers to anneal to their complementary sequences. Afterwards, DNA polymerase binds to its primers and extends the DNA sequences that are present. So instead of having two DNA strands, we end up having four. And we continue cycle after cycle until we have a large quantity of the double-stranded DNA strand that we started with. So a lot of times DNA PCR is used in order to get large quantities of a DNA sequence. in what is being popularized right now is that if you have a certain primer, you can recognize whether you are looking for a certain sequence or not, and that is what goes on in PCR tests for COVID, for example. However, what we're going to discuss here is that we want to magnify a certain sequence inside a gene. So what we do is that if we are interested in a certain region and we want to amplify this region, we heat up the first cycle, we heat up the double-stranded DNA, then we use primers that we know are going to anneal to the beginning and the end of the region we are interested in. Once they anneal, DNA polymerase is going to bind to the primer and is going to extend it, and it's going to bind to the other primer and it's going to extend it, and that way we are going to have extracted the sequence that we are interested in. This sequence is amplified over many PCR cycles. After we have a big enough quantity of DNA, we add restriction enzymes that were present in the primer sequence at the beginning, causing the sequence of interest to be cut using sticky ends. We treat a plasmid vector with the same restriction enzyme, then we incubate it with the cut region, DNA region that was amplified, and we make a plasmid vector with the DNA of interest. There are many applications to PCR, one of which that we described is the direct isolation of a specific segment of genomic DNA. So if we want to amplify a certain gene or multiple genes, we can use PCR to do so. We can use reverse transcription PCR in order to recognize what type of protein is being synthesized. When we talked about recognizing protein sequences, we talked about Edman degradation, and we said it's a very complicated process and that genomic processes are being used instead. And the genomic processes that are being used instead are reverse transcription PCR, where mRNA is extracted, then it is reverse transcribed. After it's reverse transcribed, the sequence is amplified using PCR, and the protein is identified through the DNA sequence that it has. PCR is also used to prepare probes that anneal to complementary sequence in order to recognize these sequences. Last but not least, we are going to talk about the classic method of DNA sequences. There are new DNA sequencing techniques that are a lot simpler, and some of you are going to do a project on those at the end of the semester. So the idea for DNA sequencing is that it uses PCR in order to decipher what the sequence of bases in within a certain gene of interest. So I want to know what bases make up the certain gene, so what's the sequence of bases that make up that gene. And in order to do so, we have a specific type of base. You know the traditional DNA base, which looks like this on the left, which is the deoxyribonuclease triphosphate, the DNTP. And the reason it's called DNTP is because it's a deoxyribonucleoside, so the second carbon has a missing oxygen, hence it's called deoxy. In DNA sequences, we use a special type of base, that's dideoxyribonucleoside, where not only the oxygen on the second carbon is missing, but also the oxygen on the third carbon is missing. If the oxygen on this third carbon is missing, that would prevent the formation of a phosphodiester bond later on, because as you know, this is where the following base is going to bind. So if it's missing, if the oxygen is missing, that means we cannot add any more bases to the sequence. This would shorten the length of the DNA sequence that we have if it incorporates ddNTP instead of a dNTP. So the trick here in DNA sequencing, the classic DNA sequencing, which is called the Sanger method, is if we have a certain DNA sequence, we are going to have it go 4 different PCR reactions at the same time. For a PCR reaction to take place, you need template DNA, that's the original DNA that you want to magnify. You also need dNTPs, bases. You also need a polymerase, and you need a primer. So the contents of all 4 reactions is the same, except for one difference, is that each reaction, in addition to you have, as we said, dNTPs. There are 4 types of dNTPs. There's DATP, DGTP, DTTP, and DCTP, right? We have C with G and A with T. We have all the single Ds in all the reaction. However, in each reaction, we have, in addition to that, a dideoxy ribonucleoside. And we know, for example, the first reaction has a DDATP, the second reaction has a DDGTP, this one has a DDTTP, and the last reaction has a DDCTP. So what happens, as we mentioned, in each cycle of PCR, dNTPs are incorporated and polymerized, and then we go through the next cycle. So what happens in each of these reactions that has an additional ddNTP to it is that once this DDATP is added to the growing DNA strand, the strand stops, cannot be extended anymore, because we said we are missing the oxygen on the 3rd carbon. So that would prevent the formation of a new phosphodiester bond. So the length of the DNA strand is cut short. And we know, because we added the DDATP, that each strand that is not the entire length of the gene has an A at the end of it. The same thing happened in the second reaction. However, this time we have a DDGTP. So each time a DDGTP is incorporated, the DNA strand is cut short. And because this reaction has only DDGTPs, that means that the last nucleotide in each strand is a G. And then we have a DDTTP and a DCTP reaction. This one we know all the last nucleotides are a T, and this one we know all the last nucleotides are a C. So we run these nucleotides as a gel, and we can tell at this length, let's say this is a 5 base length, the 5th one is an A, and the 2nd one is an A maybe, and the 7th one is an A, the 3rd one is a G, and so on. And at the end we're going to have the whole sequence shown up to us by deciphering the last dNTP in each DNA strand. So as I just mentioned, the ddNTPs are randomly incorporated at the position of the corresponding dNTP. This causes the polymerization process to stop at the position where the ddNTP is added. Nowadays, there are fluorescent tags included for each type of the 4 ddNTP. This helps us recognize what the last nucleotide on the truncated daughter fragment is. So we can distinguish it using fluorescent colors and fluorescent tags from other ddNTPs. So we end up getting something like this, depending on the strand's length. So this is the y-axis is the strand length, and depending on what fluorescent tag it has, we can tell which type of ddNTP we have, so we can tell what the sequence is.